If the genes and proteins of the human genome do not have to possess homologs within the human genome, then they certainly do not have to be homologous to the genes of other organisms. The genes which function in the human eye do not have to be homologous to those which function in jellyfish eyes. Human clotting factors do not have to be homologous to the immune proteins of invertebrates. The genes which control development of a zygote into a human infant do not have to be homologous to those which direct worm or fish development. The genes required for the function of the human heart and brain do not have to be homologous to the genes which are required for the function of fruit fly hearts and brains. Human cell division could use genes which are entirely different from those found in unicellular yeast. The genes which humans use to prevent cancer could be completely different from those found in nematode worms or fruit flies. If the creationist model is true, then there is no reason to expect homologous genes in organisms which are completely unrelated. If the intelligent design model is true, then there is no reason to expect that the genes required for the complex human molecular pathways could function in much simpler organisms which lack many of the components required in human pathways. The evolution model, however, claims that humans, fish, fruit flies, amphibians, worms, etc., have evolved from common ancestors. As a result, the evolution model alone predicts extensive genetic homologies between humans and other organisms. Without scientists who use the evolutionary model as a guide, our understanding of the human genome would be far less than what it is today. During the 20th century, most of the discoveries about genes which function in humans occurred after research in other organisms, such as flies, mice, and bacteria, which laid the foundation for predictions on what to expect in the human genome. For example, the Hox genes which were first discovered in flies with abnormal wing or antenna development were later shown to be critical for normal human development. In many cases, normal human genes can restore normal function to flies with mutant fly genes.